Hello again, everybody. I'm back with a short video about the first exercise in Lab 5 today. So the first exercise deals with enzymatic activity, specifically dealing with a class of enzymes called a protease. You know, what a protease does is it breaks down peptide bonds between amino acids and other proteins. So in a sense, a protease is an enzyme that breaks down proteins. Another important thing to remember about enzymes for this lab is that the three-dimensional shape of an enzyme is very important. So remember that most enzymes are made from proteins, and proteins are polymers of amino acids. So they're made of a long string of amino acids joined together by peptide bonds. Now that long string of amino acids, or polypeptide, is going to fold into a three-dimensional shape, and this three-dimensional shape is critical for the function of the enzyme. So let's say, for example, let's look here. So let's say this is an enzyme folded into its three-dimensional shape. There's a special part of the enzyme, like a pocket or a crevice, that's called the active site. So let's just say, just for example, this is the active site of this enzyme here. So you can see, so I've drawn some little square substrates around, and you can see the shape of the active site is very complementary to the substrate, so that it can bind it very easily. So as long as this enzyme can maintain this proper three-dimensional shape, it can bind substrate in its active site and catalyze a chemical reaction. However, under some conditions, enzymes will lose their proper three-dimensional shape. So say, for example, we expose this enzyme to conditions such as high heat, low pH, or high salt concentrations. That's going to unfold the protein. That's a process called denaturation, so that the protein no longer has this nice three-dimensional shape with that specific pocket called an active site. Now it's completely unfolded. The three-dimensional shape of a protein is crucial to its function. So when it loses its three-dimensional shape, it loses its active site, and it can no longer function when it's denatured or unfolded in this way. Now sometimes we can return the protein to its normal conditions, so the normal temperature it usually functions at, or the normal pH, the normal salt concentration, and many times that enzyme, that denatured protein will refold back into its normal three-dimensional shape. If it can regain its normal three-dimensional shape, it'll also regain its enzymatic activity. Okay? This is something important to remember when we look at exercise 1 of your lab. Okay, so in exercise 1, we're looking at proteases, specifically proteases that come from tropical fruits like kiwis, figs, pineapple, etc. They contain proteases, again, which are enzymes that can break the peptide bonds between the amino acids in a protein, breaking down the protein. So what you're going to do, we're going to be working with a protease from kiwi fruit. You'll be using some fresh kiwi fruit. Kiwi fruit contains a protease called actinidin. The protein that we'll be digesting is gelatin. So gelatin, this is the very same stuff that's in Jell-O. Okay, so, and it comes from an animal source. You might not realize that Jell-O is actually not vegetarian. Jell-O comes from an animal source. It actually comes from the hooves and bones of animals that are boiled down. Then when we put it back in solution, when we make Jell-O, it actually forms a cage-like structure that can hold water. Okay, and that's why Jell-O looks like Jell-O. What we'll be using is exactly the same kind of stuff that you find in Jell-O, only it's unflavored and not quite as dense as Jell-O. So let's look at the conditions. So you're going to have 4 plates with 1.4% gelatin. And we're going to put different solutions on them. Plate A, we're just going to put plain water on. This is going to be your negative control. You might be familiar with the negative control from lab number 3, when you use the different biological indicators to identify different biological molecules. Water is a negative control. So this gives us a baseline. This lets us know what a negative result looks like. B, we're going to put kiwi fruit. Now remember that your kiwi fruit contains a protease called actinitin, so it's possible that there could be proteases in your kiwi. Those proteases can break down the gelatin inside your gelatin plate. If that happens, it's going to break down that cage, and the water is going to escape, and it's going to melt the gelatin. Plate C, we're going to put kiwi, but this is kiwi that's been heated and then cooled. So remember what I said about denaturation, and think about how that might affect the results for the kiwi fruit after you've incubated this for the appropriate time in lab. Plate D contains meat tenderizer. Meat tenderizer also contains proteases. These proteases break down the proteins in the meat. That makes it more tender. So again, think about what the results for this plate might be. Finally, the last plate E, there's going to be a solution of 2.5% bacterial protease that you put on this. So you're familiar with the concept of a negative control. This is a positive control. So this is a control where we expect to see melting of gelatin. We expect a positive result. So now not only do you have a negative result to compare to the rest, now you'll know you have a baseline for a positive result as well to compare your other results to. So that's exercise 1. I will be back shortly with some short videos for exercise number 2 and exercise number 3.